Welcome to Study Loop.
We are given a scenario in which one gene results in two different proteins. How can that happen? Let's look a little at the overall process of what happens going from gene to protein. What I have drawn on the top there is a gene, and on that gene, the color coding is such that the black parts encode exons, and exons are stretches of DNA that encode usually for a particular protein domain. Introns are non-coding regions of the DNA, but they are found within the gene. UTR is the untranslated region, and in the blue is the exon, which is the same as the black, but in the RNA. So a gene gets translated to RNA. I know I'm skipping a bunch of things in the middle. We'll talk about those, okay? So gene goes to mRNA. So here I have three different kinds of mRNA that are resulting from the same gene. 1, 2, and 3. Okay, now when the gene first gets transcribed, so in transcription, we are looking right here, it gets transcribed. The resulting RNA is called pre-mature RNA. And in order for it to become mature RNA, splicing needs to happen. So splicing, okay? And splicing is the removal of the intron of the red part, the non-coding region, and the exclusion of exons that you don't want at the moment. So for example, in mRNA1, in scenario 1, we have spliced away the intron and the square exon, right? Right here, protein 1 that is resulting. So down here we have proteins. In scenario 1, we have spliced away the intron, the red part, and the square. In the second scenario, right here, we have kept both of the domains. So that is a different alternative splicing. And in scenario 3, we have spliced away the intron and the other exon, the triangle exon. So that's how you result in different kinds of protein from the same gene. And the choice that gives us the correct answer is choice C. Choice A talks about the 5' cap and the poly A tail. So let's look at what that talks about. So down here, I have the mature RNA, and this is a little bit of a more detailed view of the steps between going from pre-mRNA to mRNA. So to the pre-mRNA, you add a 5' cap, a polyadenine tail or poly A tail, and the addition of the 5' cap on this side, on the 5' region, and the poly A tail on the 3' region, they affect the stability of the DNA. It makes it more difficult for ribonucleases to get to the RNA. I'm sorry, I think I said DNA just a second ago, to RNA. And in addition to the untranslated region, which is the green part here, RNA binding proteins bind to the untranslated regions, and these also modulate the stability of the RNA. You only get mature RNA after you add the 5' cap, the poly A tail on the 3', you get the addition of the RNA binding proteins, and you have spliced away whatever introns and exons you don't want to make it there. Okay, so this is what choice A is talking about, the 5' cap and the poly A tail. And it says that one protein had a 5' cap and the poly A tail in the mRNA, and the other protein did not. So recall that the resulting of different proteins results from different splicing, and the addition of 5' caps and of 5' caps and poly A tails occur in this part. So when you go from pre-mRNA to mRNA. So you would add them here, 5' cap and the poly A tail here. Okay, so if you add those two things at this step, then that means that they will also be in all the resulting differentially spliced. So they either all have the 5' cap or none of them do. right? Because that happens before the splicing. So choice A is not correct. Choice B, one protein has a 5' untranslated region and a 3' untranslated region. Again, that's something that happens way before the step where you get different protein, the differentiation of different proteins. So the untranslated regions are present even in the DNA. They are transcribed from the beginning. So either all mRNA products have them or they all don't. Okay. Choice D. The gene produced mRNA molecules with differing stability. If that was the case, then you wouldn't observe the protein, right? Because we are being told that the gene results in two different proteins. So if one mRNA has a low stability, it's not making it to the protein level. It's being degraded before it could be translated. So the only viable choice here is choice C. The gene is alternatively spliced.